Okay, guys, so hopefully this will be coming through to you pretty quickly. I do apologize for not getting this out sooner, but unfortunately, I unexpectedly went into labor a little bit soon, and I've been trying to adjust to life with a newborn, which has been much more difficult than I predicted. Okay, so what we're going to talk about today is we're going to talk about specific host defense mechanisms, and this is more of like adaptive immunity. So whenever you guys get a vaccine, this is the type of immunity that you're going to see occurring, that what we do to kind of more fight against more sophisticated diseases, specifically viruses. Okay, so the idea here is immunology is just basically the study of the immune system and how we give our immune responses. So our primary functions of our immune system is basically what we have is we need to be able to tell what is self and what is non-self. And then we need to figure out how to get rid of that non-self. And so basically we've got these things called lymphocytes. And lymphocytes are technically white blood cells. They are a very small amount. They're going to be, remember how we talked about phagocytes before? Those were the major portion of white blood cells. Well, lymphocytes are made in the lymph nodes, and they are a smaller portion of our white blood cells. Specifically, they're about 7% or so. And what they are is they're what we call our B cells and our T cells. And then we also have natural killer cells. And basically what they do is they're responsible for our immune responses as we look at adaptive immunity and how we fight off against stuff that we can't just, ingest and destroy when it comes to phagocytosis. Okay, so within T cells, we have two different types. We have the helper T cell and the cytotoxic T cells. So the helper T cells are going to be found in the first portion here, humoral immunity. The cytotoxic T cells are going to be in cell-mediated immunity. Okay, in humoral immunity, this is what you're going to see more when you have a vaccine that you take or given, or you get sick with the flu, for instance. You're going to see something where you produce antibodies, and those antibodies are going to be stuck in your body for the rest of your life, hopefully. Like I said, sometimes you need a booster as well to continue that. But at the end of the day, they're produced by your B cells, and they're basically, their job is to go and destroy microbes. They're the ones that are get rid of and destroy all of the whatever it is that they're looking for. And they're specific to that specific microbe. So that's where you get that, what we call, permanent immunity. When we're talking about cell-mediated immunity, antibodies don't really play a huge role in this one. It's more the cytotoxic T cells that actually go through and break down and destroy the microbe. And that's because they secrete specific toxins that allow for that to happen. This is what is attacked whenever you have HIV. So this is what we're looking at when we're looking at our T cells. Yes, you still have helper T cells that are attacked as well, but what really they target is the cytotoxic T cells. Okay. So again, the major difference here, a lot of times humoral immunity, that's just going to be basically the antibodies. For here, we're mostly going to be concerned with hypertoxic T cells. We also might have some hypersensitive T cells or natural killer cells. Those are the ones that really are involved in cell-mediated immunity. Okay, so when we're talking about immunity, most of the time when we're talking about this, especially when we're talking about this type of situation, we're going to be talking about acquired immunity. And that means that we have this immunity against something because we have either been exposed to something or we've been given the inactive version of it, and we have made antibodies in order to combat this. Okay, so first is active acquired immunity. This is going to be within a person. This is going to be long-lasting immunity. So these are going to be IgG, which we're going to talk about in a little bit here. But basically what it means is that this is going to be the permanent immunity. When we talk about passive acquired immunity, usually honestly, this is one also too, this is like your vaccines. Passive acquired immunity, you might have heard with talking about COVID-19 and the different types of treatments that they're looking at for it. One of them is to give blood cell to inject blood from already immune people, so people who've already had the virus, into people that don't, or people that are sick with it, so they can get the antibodies to fight this off. This is only going to provide temporary protection because it's not going to be your actual antibodies. Now, hopefully the idea here is it goes from passive to acquired because it kind of jump starts your system and allows for you to start making those and alleviate your symptoms, so that way that you can make the active acquired immunity. But that's what a passive acquired immunity is. So that's a good example in the real world. Okay, so we have two different ways. We have natural and artificial. So natural is just you get sick. So this happens if you get the flu, or if you got the COVID-19 and you recovered from it. You now have the natural active acquired immunity. We also have artificial. So those vaccines that you get, that's going to be anything that's an artificial. So you got the flu vaccine this year. If you got a measles vaccine, all of those are going to give basically artificial immunity. Okay, so there's more different types to this, but the idea here is that you most of the time are either going to have either a live vaccine, live pathogen, or a dead pathogen. Inactive is better more to state this, because most of those pathogens aren't really living or dead to begin with, but we would have active or inactive. Okay, so the different types, the attenuated, that's going to be your live vaccines here. Okay, so this is what we're going to be concerned with. So that would be your live vaccines. An example of that, it would be like your oral poliovirus vaccine is an example of this. If you have an inactivated vaccine, that's going to be kind of the regular, that would be normal polio vaccine. And basically what it is it's inactive, so it's killed or dead or whatever it is that you're getting vaccinated from. It does not affect your body. A subunit vaccine, so that one's where you're going to have basically, the best way to put this is you got parts of it, so specifically proteins, usually the proteins that you need to make antibodies against are going to be in this subunit vaccine, and that'll be what you're vaccinated against. A conjugate vaccine is a similar situation. It's going to be one part of the vaccine, one part of it, of the virus. And then another big one is the toxoid vaccine. So that's going to be whenever you have toxins that are produced by the specific virus, and therefore you're given the toxins. So you're not really vaccinated against the virus itself. You're vaccinated against the toxin, which is what makes you sick. So therefore you have vaccination against it, but you don't have 100% vaccination, if that makes any sense. You're not vaccinated against the virus, you're vaccinated against the toxins. Okay, and the example of this would be Tdap, the Tdap vaccine. That is a toxoid vaccine. Okay, so a lot of children, you'll receive vaccines. I really just experienced this myself. My daughter received her first vaccine when she was in the hospital. And you receive vaccines all throughout your life. I still received a flu shot vaccine. So the flu shot, I still got that this year. You can still receive a vaccine well into, your 20s, 30s, 40s, 50s, 60s. And I'm sure that once they come out with a COVID-19 vaccination in the next year, a lot of our older generation will be vaccinated against the COVID-19. That'll probably be their first target, or anybody that's would be considered a target or a susceptible population. Okay, so passive acquired immunity. So an example of this, like I said, was telling you we're transferred from an immune person to a susceptible person. The other way in which you can get passive acquired immunity, this happens when we're talking about, I talk about this a lot when we talked about, especially in anatomy and physiology, when we talked about immunity. So in the third trimester, what happens is the placenta opens up and allows for antibodies from the mother's blood to go across the placenta and to reach the fetus. This also occurs too whenever you talk about breastfeeding and lactation. A lot of times we're passing more of that specific antibodies in the breast milk to the children, so that way they can be immune and have a better immunity to the baby. And so that's what just happened for me. So what happens there is that because I'm immune to the flu for this year, because I got the flu shot, my daughter is immune to the flu as well. Because I'm immune to measles, because I got the measles vaccine when I was a kid, she's immune to measles up until 9 months. And then that's when they start really upping on the vaccinations for the children. because of the fact that they're no longer immune to all the things that the mom is immune to, because of the fact that, she's losing that short-term passive acquired immunity. Okay, so when we talk about humoral immunity, the big thing we need to talk about first is going to be antigens. Okay, so this is going to be any type of foreign substance that we can, find within. A lot of times when we talk about foreign organic substance, it's going to be kind of like, either a viral protein that we can process, or, maybe possibly a polysaccharide that we can process, something along those lines. And the idea here is that we are able to go ahead and process it in an antigen presenting cell, which will typically, be, a phagocyte, or, a monophage, or something like that, macrophage or something like that. And what happens there is we're able to break it down and make it into a we're going to break it down and present it in order to get the T helper cell to start stimulating the production of our B cells, and then our B cells to produce our antibodies. And so that's essentially what this antigen is. A lot of times we look for those, and we want to make sure that whenever we have that, we'll have an antigen presenting cell. Now, I will be giving a short video in addition to this that I want you guys to look at, where I'll draw the process of an antigen processing and presentation within an antigen presenting cell. Okay, with antibodies, essentially what they are is they're proteins that are produced by the lymphocytes, and what they're kind of, we call them globular glycoproteins, but essentially what there is, you find them within a lot of different places. The colostrum that they have in the first couple days that we produce, that mothers produce, that has antibodies in it. That's the real good stuff. Your lymph obviously has it. Your blood plasma has it. Saliva, tears, those are ones because they're producing. The reason why you have saliva, partially why you have saliva, is to get rid of the bad stuff that you can either breathe in or can be found in your food that you didn't know about. Those are part of the reasons why you have saliva. It also starts the process of breaking down your sugars, but that's a whole other thing. Same thing with tears and mucous membranes. Those all have antibodies within them. Okay, so I'm going to stop here because I think we have enough time. Okay. And then we'll go ahead and actually we are still good for a couple more minutes. So let's go ahead and keep going. I apologize. I just want to make sure that we had good time. We are on 12 minutes. I try to keep my videos to about 20 minutes in length so they're able to post to YouTube, just so you guys know. Okay, so this is the basic structure of an antibody. This FC fragment is going to be considered our fragment, our constant fragment, or whatever is going to be the same for every single antibody. And then this area up here is our FAB, or it's our changing region. It's the variable region. So we have a constant region, and then we have a constant region, and we have a variable region. And those essentially change. This determines whether or not the antibody is going to be effective against the flu, or it's going to be effective against the COVID-19 virus. Okay. In addition, we also have a light chain and a heavy chain. So you have the heavy chains are over here, and our light chains are right here. Okay. So they're the shorter chains, and these are the longer chains. Okay. So this is all the amount of antibodies that you have. You obviously have antibodies that are always there. Like I said, you have IgGs. Those are going to be there for the rest of your life. And the idea behind that is that you have those will be there to help you protect against other things, but they're not going to be, the ones that, if you don't have the antigen to stimulate it, you're not going to have the same amount of antibodies than you would whenever you're about to get sick. Does this make sense? So we have to think about this type of stuff. A lot of times too, we have these are different types of antigens that we can have going on there. Okay. So what happens here? Okay. So here we're going to talk about this. This is what would happen. There's a couple of different ways that we can do this. I'm going to go ahead and write how I would like you guys to know this. But just so you know, the idea here is that you have an antigen receptor here, which is going to be what we call a what we're going to call usually it's going to be some type of TCR, That's going to attach to your antigen fragment. And then you're also going to have, in addition to that, you're going to have what we call a CD4 plus. It's going to go ahead and attach to your MCH class 2, which I'll show in a little bit here. But that's this whole thing that's happening here. What happens here, go ahead and it activates these B cells to start multiplying. You get regular B cells that are going to make your antibodies, and you get B memory cells. And essentially what happens is that they recognize this antigen again whenever it would come into your body and automatically start making this antibody. So you're essentially skipping this whole first step here. So this whole area, the next time, it's just going to remember it and start making the antibodies right away. Okay? And then we go ahead and make our plasma cells, and those plasma cells make our antibodies. Okay? So the idea here is that you go from B cells to plasma cells, and then plasma cells to antibodies. And what happens, this can take anywhere from up to 10 to 14 days to for the antibodies to be produced. A lot of times, whenever you have your initial immune response, you don't even realize that this is happening in your body. You've been sick, but you don't show any symptoms yet. You maybe like, day 3 or 4, you might show some symptoms. Okay. Or, day 5 or 6 or something like that, you'll start to show some symptoms. From there, you're already starting to use that immune response. And so therefore, you're already going to go ahead and be making antibodies, or at least trying to make antibodies. You're going to be going through the initial immune response system. If you're exposed to more antigen, okay, you're going to have, so like, let's say you get sick again. So you're exposed, you get the COVID-19, you're exposed. It takes 10 to 14 days for antibodies to be produced. You recover. Okay. Now you're immune to it. Okay. Which is what we hope for. I mean, they can't say definitively because they haven't done enough tests, but we'll just say for our purposes that you are immune to it. Okay. Now you're going to go ahead and get another exposure. So somebody else comes into your dental clinic and they're exposed to they have the COVID-19, but they don't tell anybody. Shocking, right? And then you get another exposure. This is a secondary response. The production of antibodies will actually be much quicker than this 10 to 14 days because you're already immune to it. So you won't even get past to showing symptoms. Ideally, you may get a little sick, but you're not going to get super sick. And that's the idea behind it is that you have a permanent response that's able to protect yourself against getting sick again. Okay. Again, when we look at this, again, this is just, again, there's usually what happens is that all of the B and T cells are made in the lymph nodes, but realistically, your immune responses happen in the blood. They're usually initiated by the spleen. Okay. We want to look at to make the B and T cells and all those types of stuff that you need for your immune response, they're going to go back to the lymph nodes for that. Okay. So there's 5 different types of antibodies: IgA, IgD, IgE, IgG, and IgM. Okay. So IgA, this is found in secretions. IgD, don't care about it. Cross it off. You only need to know it's an antibody. We aren't really sure what the heck it does, but it's a different type of antibody. It has a different structure, so that's that. Not really sure what goes on with this. IgE, this is going to be responsible for your allergic responses. This is long-term immunity, and this is going to be short-term immunity. So when you initially get a vaccine, you're going to make IgM, and then hopefully once the IgM starts to fall off, you'll have made IgG, and therefore you'll be immune for the rest of your life. If you need to get a booster for this vaccine, it means that over a certain amount of time, the IgG count falls off, and you need to make more IgM to make more IgG, so the way that you're able to be immune for the rest of your life. Okay? So here's some example. This is a non-secretory IgA. Most of our IgAs are going to be these ones, dimers, which means that they're found in our secretions. So don't be concerned with this picture here. Be concerned with this picture right over here. Okay? IgD don't really, again, you can see all the difference here is just this link between the different 2 heavier chains. This is the only difference between this and our IgE, so the disulfide bonds. And again, remember, these are proteins, so that's a good example. So again, IgD, IgE, IgG, and then the big one that you'll need to know is IgM. That's going to be a pentamer. And you can see how much space that takes up, that this is why it's a short-term antibody. The IgG is more of a long-term solution for you. Okay? Okay, so don't really be concerned about monoclonal antibodies. Just know that they are using for, the idea here is that they're mostly used right now for immunodiagnostic procedures. We would really mostly use them to diagnose if you have a specific type of disease or if you had a specific type of disease. So that's just the idea there. But we're also looking at different ways to use these monoclonal antibodies in the future. And that's the only idea that you need to know from here. Okay. So whenever you have an antibody antigen complex or an immune response, okay, what's going to happen here is you're going to basically activate a couple of things. You're going to make more leukocytes. So essentially, you're going to make more white blood cells. Remember, leukocytes are the same thing as white blood cells. You're going to start killing bacteria cells. And you're going to increase phagocytosis because you want to do this idea of opsonization. And what opsonization is, it's essentially just a process in which we go through and allow for more antibodies to recognize more of the antigens. And then essentially what happens is that we get more, because they've been more activated, now we've activated our phagocytes and we have increased our ability to kill things within our body. Okay? Okay, so we will stop here. And hopefully that is enough time. And then we will go ahead and pick up mediated immunity in the next one.